Good day, everyone. This is our completion lecture on our module number 4 on protozoa, algae, fungi, mosses, and words. I believe we ended with this slide on the um, discussion on fungal life cycle, similar with plants. They show alternation of not technically not alternation of generations, but we can appreciate that sexual and asexual reproduction are interconnected in fungal species. As a review, so spores that are released from mycelial network of fungal species can germinate once they land on an environment, a substrate with enough nutrients, with um, supportive environmental conditions. No? They can germinate as another set of mycelium on a different location. So asexual reproduction does not entail genetic diversity, as the spores are just clones of the parent fungal component or mycelia where it came from. No? So, in terms of sexual reproduction, in instances that are favorable, fungal species can carry out sexual reproduction that covers a number of steps. So, plasmogamy is in the initial stage where haploid cells from mating different from two mating and different mycelia form a transitional state, a heterokaryotic cell. Heterokaryotic cell possesses two or multiple nuclei. Eventually, the step or the stage of karyogamy ensues. Karyogamy is when the nuclei present in the heterokaryotic fungal cell will fuse to form a diploid zygote. So this diploid zygote is characteristically genetically different from the parents, um, who continue to develop, and when the time comes that it's able to produce haploid spores, spore formation is a product of meiosis, where genetic diversity is increased during the process. These spores later on can form a multicellular mycelium. Once germination is successful, then the cycle can just repeat, or can the both cycles of asexual and sexual reproduction can just repeat depending on the factors, no? Environmental factors to be specific. Okay. Please know, or I think I've mentioned already, that in most cases, fungal species undergo asexual reproduction, as the process is less intensive, less energy requiring. So in most cases, the default is for fungal species to carry out asexual reproduction. In terms of the different types of spores that are produced as sexual spores, let's take note of two different types. So we have sporangiospores, which are formed by successive cleavages within a sporangium, within a body, within the sporangium body. The sporangium, as you can see in this picture, is attached. No, the sporangium is attached to a stalk, a special hypha called a sporangiophore. So this sporangium is a globular structure here. Inside it, the sporangiospores are present. So these sporangiospores are initially enclosed, but can be released once the sporangium opens up or ruptures. The second type of asexual spore are conidia or conidiospores. So these spores, unlike sporangiospores, are free. They are not enclosed within a spore-bearing sac. So they develop either by pinching off on the tip of a special fertile hypa called the conidiophore, or by the segmentation of already existing vegetative hypa. This is an example. So in Mucormycota, sporangiospores are common as a sexual type of spores, while for Penicillium, for some um, non-Mucormycota fungi, like Penicillium, so here you can see conidiophore, the conidiospores on the tip of the fertile hypa, and they are free, they are naked asexual spores, these structures here. Okay. For sexual reproduction, so I already mentioned about the 3 steps. Plasmogamy, when cytoplasm fuses, plasmo, cytoplasm. So the nuclei coming from 2 different mycelia, 2 different hypa or mycelia coming from 2 different species can combine. Karyogamy, when the nuclei fuse within a heterokaryotic fungal cell. And meiosis is when haploid spores are produced. No? Sexual reproduction results in the formation of specialized spores. These specialized spores are distinct or characteristic of the different divisions. Zygospores for zygomycota, ascospores for ascomycota, and basidiospores for basidiomycota. Okay, so here are some examples. So you see. Okay. All right. In terms of the systematics of the kingdom fungi, so there are 9 major fungal phyla that are known, no, that are currently recognized. Okay, we have the Opisthoporidea, the Chytridiomycota, the Neocalymastigomycota, Blastocladiomycota, Zoopagomycota, Mucormycota, Glomeromycota, Basidiomycota, and Ascomycota. So the main traits of these major phyla are listed on the second row, the second column rather. Opisthophoridia, which includes representative genera such as the most popular one, the Nosema. They are parasitoids. They are intracellular parasitic fungi. Okay. In terms of the structural components of the cells, the mitochondria are reduced, called mitosomes, and usually they follow not usually, but representatives follow what we call an intracellular life cycle. So they have to have a host cell for them to survive. Then we have Chytridiomycota, which are zoosporic. So the zoospores have posterior flagella, and often members of chytrids are aquatic organisms. They are decomposers, some can be parasitic, and the presence of a hypa is usually rare, no? So some examples include Chytridium, Spizillomyces, no? The zoospores have flagella. Okay. Then we have Neocalymastigomycota and Blastocladiomycota, which are more less popularly known fungal phyla. Just take note of their characteristics. Okay. Then we have Zoopagomycota and Mucoromycota, which are related with Zygomycota, no? Synonymous phyla. So Zoopagomycota are mostly parasitics and symbionts of animals, or could also be parasites or symbionts of other fungi. Examples include Entomophthora and Conidiobolus. Zoopagomycota are also popular as members having the capacity to parasitize and become pathogenic to insects. So they are the common entomopathogenic and amoebophagous fungi. They eat amoeboids, no? They eat protozoans as their host cell or host organism to get their nutrients from. Then we have Mucoromycota or Mucormycota, which are saprotrophic and usually plant associated, found in soil, mostly in soil, no? Mucormycota are classified or characterized as having non-septated hyphae and the formation of the zygospore as a sexual spore. Examples include your common bread mold, the Rhizopus species, and mucor. No? These mucoromycota are fast-growing. No? Common asexual reproduction. Commonly, they carry out asexual reproduction so they can thrive. And sexual reproduction is via the production of zygospores. As mentioned, they are mostly terrestrial fungi. There we have glomeromycota, the definitive characteristic or description of glomeromycota are, they are what we call the arbuscular mycorrhizae fungi associated with plants. They are obligate biotrophs, they are obligate symbionts, no? They are observed to follow asexual cycles only, no observed sexual reproduction to date. Okay? Then the basidiomycota are the ones that produce basidiospores on their specialized club-shaped structures called basidia. Included here are mushrooms, smuts, and the typical filamentous basidiomycetes. No? Examples include agaricus, Pachynia, and Ustilago as representative genera. Then we have Ascomycota, the sexual spore, the ascospore, is produced inside a sac-like structure called the ascus or the asci. No? They have diverse forms ranging from unicellular yeasts to cup fungi. So we have Saccharomyces, also Ascomycota are popular because these fungal genera can also cause infections in plants. No? Many of them are plant pathogens. For example, representative genera under Ascomycota are Saccharomyces, which is a yeast, Aspergillus, which is a mold, and also Neurospora. No? Now let's take a look more examples on fungal representatives. Under Chytridiomycota, the chytrids, they are fungal species predominantly aquatic, can also be found in soil and also in freshwater bodies such as ponds and streams. They can also be found in ecosystems with low water activity such as desert soils, no? And also as associated microbiota in animals such as ruminants. No? They are distinguished from other fungi primarily because of their characteristic motile cells. No? Zoospores and gametes are characteristically motile. And most of them have a single posterior flagellum. You can see here the whiplash flagellum of a zoospore, allowing it to be motile. Okay? Then we have division Zygomycota, the zygomycetes, the poster genera or species under Zygomycota is Rhizopus stolonifer, no? Which is the common bread mold. Characteristics include formation of zygospores, sexual zygospores. They develop within sporangia, zygosporangia, no? In species that are able to reproduce or to manifest sexual reproductive cycle. Sexual reproduction in Zygomycota will require two physiologically distinct mycelia, so designated usually as a positive, a plus or minus strain coming from very different mycelia. Therefore, no, combining their characteristics in the progeny, or the progeny fungi with combined characteristics. Then we have division Basidiomycota, the club fungi. Best example of Basidiomycota are mushrooms. The basidium is a club-shaped structure produced at the tip of the hypha of the fungi, and inside it are basidiospores. The basidia may be held within fruiting bodies called basidiocarp. So here is an example of a basidiocarp, no? Where basidia are held. Okay, so more appearance underneath it, the gill structure, the gilled area where your basidia are present, and the basidiospores are can be extracted from here. So you can see the basidia and then the spores colored in red. Okay. Then we have sac fungi division Ascomycota. So the term or the name of the division, the group, is coming from the fact that the structure forming the sexual spores is a sac-like structure. So that sac-like structure is called the ascus or the asci. It's common in ascomycetes that asexual reproduction takes place by means of conidiospore formation. And as already shared with you, asexual reproduction is the default, no? It's a normative cycle followed by fungi. Examples of Ascomycota include Aspergillus and Penicillium as mold examples, and Saccharomyces as a yeast example. Take note of some specific structures: the conidiospores, naked asexual spores; the conidiophores, specialized fertile hypa, bearing the structure that the conidia, which produces the conidiospores. Then we have the division Deuteromycota. Although it's not really a valid group anymore, we use it as a placeholder, as a holding tank of some sort. Say, division Deuteromycota, the main purpose of this group is to house fungal species that are described in which only a sexual reproduction has been observed and no sexual stage has been observed yet. So once that the species has been identified to have a sexual stage, it can now be reclassified. So it's not really later on. The idea is later on, Deuteromycota will be dissolved. No? Once everything inside it or under it will be classified to have a sexual stage. Okay. Right. So that ends our discussion on fungi, no? Basic information that you have to know about the systematics of fungi. Now let's cover mosses and worts. Although, let's see, might be too much already. I'll cut short the recording so that it will be more consumable on your end. Okay?